Okay, welcome back. This is our fifth lesson in our Unit 1 LEAP remediation. This will be the final lesson for Unit 1. And so after this, we'll move on to Unit 2, which is genetics. But the last thing we need to talk about before we move on are proteins. And we talked about how proteins are made, but we also need to learn different types of proteins, and we need to learn what their function is in the body. And so there are 4 major types of protein in living things. And the first one is called a structural protein. And so a structural protein is for building tissues and muscles. So examples of structural proteins would be actin and myosin. And you might remember when we learned about the kids with DMD, we learned about actin and myosin. Duchenne muscular dystrophy, when we were learning about muscles, actin and myosin are muscle fibers or muscle proteins that make up our muscles. So that's just 2 examples. There's lots of different types of structural proteins. There are also transport proteins, which carry substances through the body. Of the organism. So an example of a transport protein would be hemoglobin, which we talked about in our last lesson. We talked about how normal hemoglobin is round, but the hemoglobin in people with sickle cell anemia is, I may not have told y'all that in the last video. Let me tell y'all that now. So remember this, remember this from the last video when we were talking about hemoglobin in normal adults and hemoglobin in people with sickle cell anemia. And so I don't know if I told y'all, but sickle cell anemia results in red blood cells that are shaped funny. So normally red blood cells are perfectly little round shaped structures, but if you have sickle cell, your red blood cells are kind of shaped like a crescent roll or like a crescent moon. And so because they're shaped weird, they get stuck in the arteries and the veins as they're trying to travel through the body. And so they don't transport oxygen the way they're supposed to. So anyway, one of those proteins that are called transport proteins is hemoglobin, which is found in your blood, and it carries oxygen in the blood. Okay, now an enzyme, which we're going to talk about a little bit more in depth, but an enzyme is it aids in chemical reactions by speeding them up or making them go faster. That's basically the same thing, by speeding them up. Okay, so normally without enzymes, chemical reactions can sometimes go too slowly to be effective. And so enzymes come in and they help speed up those chemical reactions that are going on inside our body, like when we're trying to digest our food, or when we are trying to move oxygen in and carbon dioxide out. All those different types of reactions that are going on are too slow. And so enzymes, which are a type of protein, help this process. And so an example, 2 examples, one is called lactase, and you may have heard of lactose intolerance. The enzyme lactase is what helps to break down lactose. And so if you're lactose intolerant, you have a problem with this particular protein. Another one is called amylase, and you'll notice that they both end in ASE, and that's because typically, not always, but typically enzymes are named with ASE on the end. And so both of these are involved with digestion of food. And then the last type of protein is called a channel protein, and its function is to move substances in and out of cells. And so a channel protein, one that we talked about in class, we made that. If you were in my class, we made that channel protein. Remember, that could open and close, fold. We folded little pieces of paper. And so that's an example of a channel protein. Okay, so in order to work correctly, each protein has a very specific structure and shape. So we know that proteins are made of amino acids, and we know that from our last video that the amino acids have to be hooked together in just the right order. And that order is determined by the code from the DNA. And but also proteins fold. So they're made into these long chains of amino acids, but they don't stay a long chain. They fold up, and they don't just fold up randomly like crumpling up a piece of paper. They have a very distinctive folding pattern, and they have to have a specific shape. They also need the right conditions. They have to have the right temperature, the right amount of water, the right amount of salt to keep their shape. If they experience conditions outside of these parameters, they can break down, and that's called denaturing. So proteins have to have very specific conditions, and different proteins have different conditions. They have to have very specific conditions and a very specific shape. Okay, so we can see the importance of that when we look at enzymes. So we said enzymes are one of the 4 different major types of proteins. And we know that enzymes help chemical reactions. And so we're going to look at an example of how an enzyme works by looking at a lactase example. And so the enzyme is lactase, and the substrate is lactose. And you know that lactose is found in a lot of different dairy products like cheese and milk and stuff like that. Sour cream, cottage cheese, all that kind of stuff has lactose in it. And so whenever you consume lactose, you have an enzyme called lactase that helps to break it down. And so this is how that happens. So your substrate, which is this green shape here, would be your lactose. So the substrate is whatever the enzyme is going to act upon, okay? And so the gray shape here is your enzyme. And so that is in this example, that is your lactase. So here's your enzyme, lactase, and this is called the active site. And look at that distinctive shape of the active site. It kind of looks like a puzzle piece. And what's interesting more so is that the substrate, which in this case would be lactose, has a very similar shape to the active site on the enzyme. And that is on purpose. It's like a lock and key. It fits perfectly. So a random molecule cannot connect to this enzyme. Only the specific substrate that this enzyme is supposed to connect with. So in this case, the enzyme lactase connects to the substrate lactose. And when the enzyme and the substrate connect, the enzyme changes shape slightly, okay, as the substrate binds. So it kind of absorbs the substrate in and changes shape. And then the enzyme either connects two pieces together or breaks two pieces apart. And in this case, the enzyme is going to break the lactose apart into two separate pieces. So this is part of the digestive process, breaking down the lactose, and then it releases the products. So your substrate is changed and broken down and then released by the enzyme. And so these enzymes are not changed in the process. Once the enzyme releases the products, it goes back to its original shape and it can be reused over and over and over again. And so another lactose molecule can now bind to this same enzyme, and it just keeps going through that same process over and over again. And the enzyme will keep on working indefinitely unless it gets denatured. And if you'll remember, we did a lab on enzymes and we saw what happens when enzymes are denatured due to acid, which puts it in a pH that's way too low, or from heating it, like when the temperature is high, that denatures the enzymes and they don't work anymore. They don't do what they're supposed to do. Okay, so if you look at this graph, you'll see how an enzyme speeds up these chemical reactions. So in a chemical reaction, you always have reactants. That's what you start with. And then you have products is what you end with. So your reactants react, and then they produce the products. Okay, so this... phone call in the front office. You have a phone call in the front office. Okay, so this is the amount of energy required for the reaction to take place. Okay, so this is your reaction proceeding over time, and this is the amount of energy required to let the reaction proceed. Okay, so here's your reactants. So the red line shows how much energy is required without the enzyme. So see how this reaction is trying to proceed, but it needs all this extra energy without the enzyme in order to produce the products, okay? But with the enzyme, your blue line here, with the enzyme, it's a lot less energy required to produce the same products, okay? And so with this particular reaction, we have an exothermic reaction because more energy is required. The reactants are at a higher energy, and then more energy is required, and the products are at a lower energy level. So that means heat is going to be given off in this reaction, because the heat of the reactants, the energy of the reactants is higher than the energy of the products. If the products were up here and the reactants down here, or the reactants down here and the products up here, that would be an endothermic reaction and heat would be absorbed. Okay, but the big deal here is the activation energy, and you can see that it requires a lot more energy without the enzyme than it does with the enzyme. And in this way, the enzymes help our bodies to do these chemical reactions without requiring as much energy on our part. And, you know, we have to have food for our energy. So, and we want to conserve energy whenever possible. And so these enzymes help us to do these reactions. Okay, well, that's it for Unit 1, Molecules to Organisms. And so we have learned, let's just do a real quick recap on what we have learned. We have learned about all the smallest units of biological things being the molecules of life. Those are not the units, but the smallest units are the cells, but the molecules of life that make up the living things are called macromolecules. And there's 4 types: proteins, carbohydrates, lipids, and nucleic acids. And then we went into a little more. They're all made up of carbon, hydrogen, and oxygen. And then some of them have phosphorus and nitrogen as well. Then we went into more detail about the nucleic acids, and those are DNA and RNA. And we learned some of the differences and similarities between DNA and RNA. And then we talked about how DNA and RNA are involved in making proteins and protein synthesis. And so we learned that protein synthesis is a 2-step process: transcription and translation. Transcription is where DNA makes RNA, and translation is where RNA goes to the ribosome and the protein is built. Then we did a lot of practice with DNA replication. Remember, you've got to memorize your coding, your base pair rule. Apple in the tree, car in the garage only applies to DNA replication. So that's when you're making a copy of DNA and you're going from DNA to DNA. And then we did some practice with protein synthesis, and the first step being transcription. And this is where DNA makes messenger RNA, and your code is a little bit different because RNA doesn't have thymine. RNA has uracil instead, so don't forget about that. And then we learned how to translate the codons. Every 3 letters is a codon in messenger RNA into an amino acid using our codon chart. We did some practice with that. We did some leap style questions with that. Then we talked about the different types of mutations. And there are 3 types: substitutions, insertions, and deletions. We learned that there are 3 major causes of mutations, one of those being errors during DNA replication, and mutagens, which are things out in the environment like X-rays and radiation from the sun, harmful chemicals, pollutants, and viruses. We also learned that mutations are not always bad. Sometimes they are neutral, and sometimes they're actually beneficial. And then we just finished up by talking about the different types of proteins and how enzymes work to help our body. Okay, that's Unit 1. So Unit 2, like I said, is going to be our genetics unit. So I'll see you back in our next video. Thank you for your attention.